Hey guys, out there in cyberspace. I've got an awesome lecture for you because we're starting in on the skeletal system. So, you're going to learn all the bones of the body and all the cool little features and bumps and holes and etc. of the body. But before we get into what each bone is and what the parts of it are, let's zoom into what it's made of. What makes a bone a bone? And it's fascinating. Not only the anatomy, the histology, the microscopic anatomy of bone, as well as the gross anatomy of bone, but also the physiology. I mean, how our bodies work. Homeostasis is about every part of our body, including our bones. And you'll learn in this lecture why exercise is so important for a healthy skeletal system. So let's get started. Okay, so let's get started with just a general look at the structure of a typical bone. We'll take a long bone. This is a long bone. This is a femur from your thigh. Well, not your thigh, but somebody's thigh, which then became a plastic cast. And let's take the basic anatomy of this, because if you were to slice this thing in half sagittally and look inside, you'd see it's not all uniform. And in fact, it's mostly hollow. And that's really important because having kind of hollowish bones is an adaptation for them to be light so that we can get around and be mobile, yet super strong. And the best engineers, when they're engineering like a bridge, what do? Well, you might have remembered if you ever took like engineering in high school and had to build those popsicle stick bridges. They make trusses, right? They make little beams that crisscross and make diagonals, and that makes the bridge light yet extremely sturdy. And it turns out evolution already figured that out for we organisms. We actually have that generalized structure within our bones. So what I have here on the screen for you is a long bone, the same bone. This is a femur. So I'm going to draw an arrow up here so you can see here's the nice hollow part. That is called the medullary cavity. Medullary cavity. And notice that up here, they also called it the marrow cavity. So, of course, in anatomy, everything has 10,000 frickin' names. Well, I'm only holding you responsible to call it what I'm teaching you it's called. So that's the medullary cavity. Now, notice that the outside of the bone is really, really hard, and that is called compact bone. So let's change colors over here, and you'll see that that's called the compact bone. So the outside is made up of substance, which is cells plus matrix, because remember, bone is a kind of connective tissue. Remember that? And so that is compact bone, and we're going to go over the intricacies of the structure of compact bone. Fascinating stuff and so beautiful under a microscope. So, but right at the very, very outside of the bone, even covering the compact bone, is something called the periosteum. So let's circle that right there. So right here, it's just the very outer part here. That's called periosteum, and of course it surrounds the entire bone. But on the very, very outside at the end, so I'm going to kind of just go like this, we have our articular cartilage. And see how good your memory is from our tissues lectures. What kind of cartilage is that? Dun, dun, dun. Doth my ears deceive me? Did you say hyaline cartilage? If you did, you are correct. That is hyaline cartilage. And we're going to go back a review over the structure of hyaline cartilage because it's so important to the skeletal system. And in fact, when you look at the ends of the bone, it looks like there's only a little bit. Yet, if it weren't for hyaline cartilage, there would be no bone. We all start off with cartilaginous skeletons. Now, we're also going to be talking about this structure right here and right here that's in long bones. That is your growth plate, better known as your epiphyseal plate. Let's spell that out. Epiphyseal plate. Your growth plate. And you're going to learn in this lecture about exactly how long bones grow, what is the process by which those little plates, which are actually, when you're young, made out of cartilage, what's the process by which those allow bones to grow. Now, changing colors once again here, notice that in addition to compact bone, we have another kind of bone. Now, in this diagram, it's called cancellous bone. I rarely hear anybody call it cancellous bone anymore. We call this spongy bone because it looks like a sponge. Spongy bone. And so we're going to do some histology and learn about the very cool things that make up spongy bone. And that's where I mentioned these like triangular trusses that you'd have in a bridge. That's what these are. They're just beams that are keeping this thing strong yet light. Now notice that, let me go back and, well, let's go and take this long bone and notice that it can be kind of divided into three main parts. So at the head of the bone, so in the case of a femur, that's up here, this is called the epiphysis head. So the ends of the long bone are called the epiphyses. So this is, remember epi means upon. So this is the epiphysis head. Epiphysis head. So that's a head. The other end is just the epiphysis. And the shaft, the middle of it, is called the diaphysis. Okay, so this is the diaphysis, epiphysis head. All right, so that's the general structural gross anatomy of a long bone cut in half. Now we're going to break it down and look at the nitty-gritty and take a look at that hyaline cartilage that we've been talking about. So let me go back here and remind you that we're talking about hyaline cartilage. So this is so cool because all life starts out from the same basic components. So you guys have heard of things called stem cells, right? Stem cells. What do stem cells do? Stem cells are undifferentiated. They haven't become what they're going to become yet. They're like new. And depending on what tissue they're in, genes will get activated that will tell those stem cells what kinds of cells to become. Well, when you're a little itty bitty embryo, we actually have stem cells. I mean, that's what we basically consist of early enough along. And you have stem cells that are going to produce cartilage. Hey, so you know what stem cells are. But it turns out that stem cells that are going to become cartilage cells or going to become bone cells have themselves their own special name, of course. And we're going to call these mesenchymal cells. So let's draw a little arrow and write this out. Mesenchymal. Isn't that a cool word? Mesenchymal cells. Mesenchymal cells. And these cells are going to give rise to the cells that will produce cartilage. Now, you guys remember this from studying tissues, right? Let's write this down. Chondroblasts. You remember chondroblasts? Of course you do. Chondroblasts. You remember what the prefix chondro means, right? It means cartilage. So these are the cells that produce cartilage. Now, remember that cartilage is a type of connective tissue, and in fact, it's a type of structural connective tissue, along with bone being another type of structural connective tissue. So you have bone and you have cartilage, and really those start out as one and the same because when our skeletons are developing, it's cartilage and then will become skeleton. So chondroblasts are actually kinds of fibroblasts. So they're kinds of fibroblasts. And they're basically, well, let's see, you see them here in this picture, right? So where are the chondroblasts here? So let's point out the chondroblasts. These little things, just these little itty bitty things right here that are shown. It's shown in kind of dark stain right here, like kind of a black. Those are chondroblasts. Chondroblasts are the stem cells that have become cartilage cells, so they're no longer stem cells, but they are by themselves before they've started actually producing the cartilage. But then they do their magic, and all of a sudden they're going to start producing ground substance. You remember ground substance? Of course you do. Ground substance plus fibers. And you'll remember that ground substance consists of like solutes, minerals, salts, things like that. And the fibers, what kind of fiber are we generally talking about here when we're talking about hyaline cartilage? You'll remember we're mainly talking about collagen fibers. But these collagen fibers are kind of invisible under a light microscope. And that means that the hyaline cartilage has this glassy look to it. So that's what all this stuff here is, right? All that stuff around the cells That's glassy ground substance and collagen fibers all around. So basically these cells are going to start producing they're going to start producing ground substance and collagen, which will just ooze out, and eventually you're going to end up with the chondrocyte in its own little space. So let's keep drawing that chondrocyte in pink. So here's our little chondrocyte with its nucleus. But now it's gotten a nice little space around it, and all around that is that ground substance, that matrix. And that matrix is hyaline cartilage. That's what that is right there. Hyaline cartilage. So why does it get a space around it? You know, it has all this space here. Well, these cells are alive, and when they're alive, they have to be able to be delivered nutrients, right? They have to live, so they have to be able to get water, and they have to be able to get nutrients, and they have to be able to have their waste taken away by the waste guy, right? But there's not any blood vessels in cartilage. You might remember that. Let's make a note of that here. Let's write that in red. Hyaline cartilage. No blood vessels. There are no blood vessels in cartilage. So if there's no blood vessels and these chondroblasts in their spaces are stuck there, how do they get the nutrients to them? Well, I am so glad you asked that question because it's really fascinating. So these guys have no blood vessels. So let's go down here and take a look at exactly what happens with these guys. Well, let's change the color to orange. It's all about this ground substance here. Ground substance of cartilage is hydrophilic. Do you remember that word, hydrophilic? Of course you do. Water, love. These guys love water. This ground substance loves water, and so because it's hydrophilic, water is going to constantly be oozing into this kind of semi-solid matrix here. So water likes to go in. H2O loves to go into the ground substance, and therefore these cells are going to take up water and nutrients through the process of diffusion. Remember diffusion, where solutes, molecules, go from an area where they're more greatly concentrated to an area where they're lesser concentrated. So what we end up having thanks to this water is water and molecules are more greatly concentrated in the matrix than in the chondrocytes, and therefore that stuff moves in. Oh, what did I just say there? I said chondrocytes. You're like, I thought you said that was called a chondroblast. Well, guess what? Once that chondroblast has made its little house, because it's barfed out all that matrix all around it and made its little space in there, it is no longer called a chondroblast, because now it is surrounded by ground matrix. It is now called a chondrocyte. This is now a chondrocyte. That is the chondroblast plus its ground matrix. And the little house, you might recall, that this chondrocyte is in, that space right there, all that space, that is called the lacuna. Lacuna matata. That is called the lacuna, or lacunae for plural. So this is basic anatomy of hyaline cartilage. So you see that up here. Now this is going to be very, very important, this hyaline cartilage, because this is going to be what ultimately is the scaffold for us to create bone when we are little itty bitty babies in our mommy's tummies. So really, really important that we understand the basic structure of hyaline cartilage. Now remember the other types of cartilage that we talked about, elastic cartilage and fibrocartilage. Well, those are all important cartilages, but when we're talking about the skeletal system, we are really focused initially on hyaline cartilage. We'll also be interested in fibrocartilage later on, but for now, make sure you understand how hyaline cartilage is produced and what its characteristics are. Okay, so before we go on, I found a couple of really awesome 3D computer animation videos that will introduce you to the basics of bone biology and really get into the nitty-gritty of osteoblasts and osteoclasts, which you'll learn about in a minute, and their physiology. So let's watch a quick one on just an introduction to bone, and this will give you a nice overview of everything we're going to be going over. So we've done a little review of hyaline cartilage tissue, but now we're going to talk about bone tissue. So here's a little intro.
The human skeleton gives the body its shape and provides physical support for the systems contained within. It also forms part of the musculoskeletal system that enables us to move. The structure of bone is optimized so that it is strong but relatively lightweight. The interior of bone is composed of bone marrow. It is surrounded by two major types of bone tissue: cortical bone, or the hard outer shell of bone, and trabecular bone, the spongy-looking center. The amount of each type of tissue in bone is dependent on the function of that bone. The basic unit of cortical or compact bone is the osteon. It is composed of successive concentric lamellae. This structure contributes to bone strength by resisting bending. Cells called osteocytes are distributed within the concentric lamellae. Osteocytes form a complex network that is thought to be important in maintaining the viability and structural integrity of bone. At the center of the osteon is the Haversian canal.
Also called the central canal.
These canals contain blood vessels and nerves. The blood vessels within bone facilitate the exchange between osteocytes and the blood. Trabecular bone is present in the interior of some bones and resists compression. Osteocytes are also contained within its structure and again play an important role in sensing local changes in strain. Trabeculae are covered in a layer of flattened lining cells that are thought to be involved in the dynamic process by which bone is formed and broken down. Bone marrow is found within the interior of bones. The surrounding trabeculae and vascular network provide structural support, nutrition, and a waste removal system for the heterogeneous group of cells found within this space. Bone marrow is a site for hematopoiesis, the process by which the cellular components of blood are formed.
Wasn't that cool? Those videos are done by Amgen. So cool, because that really gives you a three-dimensional picture of what's really going on there. Let's move on. I noticed they use some terms that we're going to be using some different terms, because again, in anatomy you have a million terms for the same thing. But actually what they're trying to do is get away from the use of proper nouns in anatomical terminology and go with more kind of generic, generalized terms. So for example, instead of the Haversian canal, it's going to be central canal, etc. So let's get started. And recall that bone is a kind of connective tissue. So bone is a kind of connective tissue. It's a structural connective tissue just like the cartilage. Structural connective tissue. And it comes in two main types. So we are going to be talking about compact bone. And we're going to be talking about spongy bone. So now we're moving into the histology. And we're going to start with compact bone. That's the hard part. That's the hard substance that makes bones hard. And so nothing more beautiful to look at under a microscope than a cross-section of some compact bone tissue. And here it is right here. I mean, look at how beautiful this is. Each one of these units here, this beautiful thing here, oh my gosh, how pretty is this? You know, what other kind of tissue is so easily recognizable as compact bone tissue? This right here is the basic unit of compact bone, and it's called an osteon. Osteon. Know that word because it's so wonderful. And there's so many components to it. So we're going to learn about all these components. Well, let's just start off with that thing we've already talked about, the central canal. That's this thing in the middle. I'm just going to color it in. So this is your central canal. And the central canal is very important to bone because whereas cartilage tissue was avascular, it did not have blood vessels, bone is vascular. we got cells in there and we have to feed them, and diffusion's not going to work for the cells of bone like it did for cartilage because it's hard bone. You can't just diffuse water across it like you could with the cartilage. So it's really important that it's vascular, and in fact, it's going to need a lot of blood, and it's producing a lot of waste that has to be taken away in the blood because bone is a busy place. Who'd have thunk it, right? You're looking at a bone and you're like, okay, it's not doing much, it's a bone. You would be amazed at the constant activity going on inside your bone tissue. The physiology is fascinating, and we're going to learn all about it. So take a look here. Here is a diagram of a piece of an osteon, and in fact, these pieces exist all through the place. So notice like here and here, you actually have kind of partial osteons. Those are called interstitial osteons. But so what I'm looking at here is kind of a piece of an osteon, and so here would be part of my central canal. And so notice that in my central canal, I'm going to have blood vessels. Okay, so those right here. So we have all these right here. Those are our blood vessels. And those blood vessels are going to consist of an artery and a vein. And arteries take blood towards the source. Veins take it away. So the artery is going to be delivering oxygen, nutrients, minerals, things that these bone cells are going to need. The vein is going to take the waste products of the bone cells away. And they're also going to be delivering hormones, which, as you'll find out later, are very, very important to the functioning of bone. Now, the other thing that you're going to find in your central canal is a nerve. Because obviously, to make your bones do what they need to do, you have to have some kind of chemical electricity in there. And that is carried through the nervous system via a nerve. So let's write that out. Our central canal is right in the middle of an osteon, and it's going to include blood vessels, artery and vein, as well as a nerve cell. So that's what you find in an osteon. Now, notice that, and actually I'll just stick with the yellow here. Notice that there's all these rings. And I'm kind of not tracing these properly, but just to give you an idea, you can see them out on these other ones here. All these rings. So an osteon is ring, 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 ring around the rosy, because that's kind of how the bone grows. It grows like this. It's like a tree in a sense. And so these rings each have a name, of course, as everything does in anatomy. Each of these rings is called a lamella. Lamella, or lamellae for plural. And along the edges of each of these lamella, you'll find the bone cells. And you'll also find little bitty tunnels. So I don't know if you can see these in this picture, but you can certainly see them here. So imagine that each of these, let me go back to the same color here. Notice that each of these in the diagram is a lamella. Okay, lamella, lamella. But then you have these beautiful little tunnels, little canals, kind of crisscrossing each lamella. See that? Those are beautiful, and those are called canaliculi. Canaliculi. They're little canals. Canaliculi. And that is really important because what good would your blood vessels be and the blood be if it's just stuck in the central canal? Little canaliculi that are going to take the blood from the central canal and ooze it. So let's go over here and just kind of illustrate that we have cells all over the place. And you can see them in the diagrams. They look like these, in the photograph, they actually look like these dark little areas. That's contains your bone cells. And we'll talk a lot about bone cells. And let me go and, of course, add in our little caniculi, our crisscrossing these areas here. So that's a generalized osteon. So bone cells have a name, and they are called, when they're in their spaces, they're called osteocytes. So you'll see kind of a pattern here. You already learned about hyaline cartilage, so you had chondroblasts that barfed out the cartilage matrix, and in the process formed a little space called a lacuna, and then became chondrocytes. Well, bones do the same thing. Okay, so with cartilage, we had mesenchymal cells, however you want to say it, that were stem cells that became chondroblasts that barfed out a ground substance with fibers, a matrix that created the cartilage, and then became chondrocytes in their own lacuna space. Well, with bone cells, it's the same general idea because they're the same general thing. They're just kind of bone is more mature than cartilage in a sense. They're going to start out with in stem cell format called mesenchymal cells. See a pattern here. So these guys are mesenchymal cells, which are stem cells. And if the right genes are activated and the right activities are activated, these guys will become little baby osteoblasts. Little baby osteoblasts. Osteoblasts are going to create bone matrix, ground substance plus fibers. And when they do so, just like what happened before, so here's your little osteoblast, and he's going to barf out bone matrix, which is basically calcium phosphate, is the mineral that we're talking about, along with some other stuff. And in the process, make himself a little space. And guess what that space is called? Just like in the cartilage, that's a lacuna. So it's an osteoblast. I'll make the osteoblast in green too. Okay, so it's the osteoblast inside a lacuna, and together we call that baby an osteocyte. And at that point, that osteocyte is kind of trapped. He is in a hard bony matrix, and that bony matrix consists of a ground substance that is going to contain a lot of calcium phosphate. So you know when your mama says drink your milk because you need the calcium? Well, you do. Your bones are a depository, a reservoir for calcium. And whenever other parts of your body need calcium, guess where they're going to go for it if they can't get it through your diet? They're going to take it off your bones. We're going to talk about that soon. So I think you kind of get the idea there of what an osteocyte is, what an osteoblast is, versus the cartilage side of things where you have a chondroblast and a chondrocyte. Both like to sit in a little space called the lacuna. But the difference is osteocytes fed by blood. Chondrocytes fed by diffusion. That's an important distinction there. So that was compact bone. Now we are on to the other kind called spongy bone. Yes, I changed my yellow there. I didn't like the other yellow. It was kind of lame. Okay, so spongy bone is what you find on the insides of bone. Now keep in mind you have different kinds of bones, right? We have long bones and we have flat bones like what you'd find in the bones of your head. And they look very, very different. I mean, flat bone is not going to have this big medullary cavity like long bones do. So let's take a note of that just off in the corner here. So we have long bones. These are two types of bones, and we have flat bones. So long bones are your humerus and your radius and ulna, your femur, your tibia and fibula, whereas your flat bones are like your parietal bones, your frontal bone. You know, most of the bones of the skull actually are flat bones. Your scapula, you know, would be a flat bone. Now, back to spongy bones. So if we were to take this long bone and slice him sagittally and look inside, you're going to see all this kind of beams, and it looks just like a sponge. So that's what we see here. And so what we're going to do is we're going to take a little section right here. Boom, ba-doom, 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 ba-doom. I love when they do this. And we're going to look at the histology of spongy bone. So what you see here is very interesting. So if you look over here, all this stuff in the middle, all this right there, it actually has, lo and behold, things that look just like hyaline cartilage. It looks like hyaline cartilage. But what makes you know that this is spongy bone is all of these bony things right here. Look at that. And that over there. I should be using, let me switch and use a different color here so we don't get confused. All this stuff here and here and here. This is cool stuff. It's bony. And in English, it means beams. And the word is trabeculae. So these are trabeculae. They're beams. These are the crisscrossing stuff that the stuff is made of. This is what is going to give our bones the property of being lightweight yet strong. They can take a lot of weight on them because of these crisscrossing beams. Basics of engineering, right? Evolution figured it out. Okay, so check this out. This right here, this is actually an electron micrograph. So take it with an electron microscope. Those babies are really expensive. Take it with an electron microscope of these trabeculae. How cool is that? I mean, look, they're just crisscross, crisscross, crisscross with lots of space in between. So, and these trabeculae are actually coated with a cellular layer, so a flat cellular layer, cellular coat, and that is called the endoosteum, which is very interesting because you remember the outside, covering the outside of compact bone was the periosteum. Peri means around. The inside of the inside is the endoosteum. Endo means on the inside. So that's the basics and the histology of compact bone and spongy bone. So what we're going to do now is take a look at the cells themselves, the osteocytes, meaning the osteoblasts, and another kind of bone cell called osteoclasts. Okay, so now we're going to talk about the different kinds of bone cells in more detail. So we have what you see over in this picture right here. These guys are osteoblasts. And isn't that a cool picture? In this picture, these osteoblasts have been stained with green fluorescent protein by injecting a gene from the glowing jellyfish. Isn't that so cool? Love science, because I'm a nerd. Anyway, those are osteoblasts. This guy over here, and we see a diagram of him up here. I mean, he looks like a multi-eyed monster, right? He looks like something from, I don't know, some Sesame Street scary thing. That is an osteoclast. So we have osteoblasts and osteoclasts, and you can kind of think of these guys as sort of opposites from each other in their action. Osteoclasts, think C for class, crush bone. They crush bone. Huh. They crush bone. I mean, look at him. He looks like he would crush bone, doesn't he? Versus we have our osteoblasts. So you say build for B. They build bone. So osteoblasts build bone, osteoclasts crush bone. And the way they do this is absolutely fascinating. And all this has to do with what's called bone homeostasis. You guys know homeostasis, right? It's the way our body maintains a balance. And so everything from pH to water to, hydrogen ion concentration in the blood, all of this is homeostasis. It's a seesaw. It's a constant battling act. And so remember that bone is calcium phosphate. That's one of the principal minerals of it. Well, your body needs calcium. And when you don't get enough of it from your diet, as we said, your body's going to break down bone to release that calcium phosphate into the blood. And who does the breaking down? Dun, 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 it's our friends, the osteoclasts. They crush bone. And so what they're going to do is they're going to secrete acid. They secrete acid actually onto the surface of the bone. So these guys are kind of crawling along. Notice that they are multinucleated. They got lots of nuclei. Multinucleated. Osteoblasts are not. So, I mean, these guys look like they're going to crush bone. They look like they're on a mission. And they're going to crawl along here. They're actually start out from monocytes, from macrophage cells. And you're like, I thought that was part of the immune system. Well, yeah. And what do macrophages do? They eat things, right? Well, these guys, these osteoclasts, are going to come along and they're going to eat things. They're going to eat bone. And they come along and they actually kind of make a little seal on the bone and they secrete acid. And if you've ever taken acid and put it on like calcium carbonate, for example, like a shell, you'll notice that it dissolves that calcium. And so that's what's going on here. These guys are secreting acid, and that dissolves the minerals off of the bone and releases those minerals, that calcium, into the bloodstream. And we're going to watch an awesome Amgen video that illustrates exactly this process right here. So these guys release calcium. Release calcium. Remember, calcium mineral there, into the blood. And that process is called resorption. So through the process of resorption, osteoclasts go along and they are going to basically dissolve the calcium from the hard bone, from the compact bone, and release it, or from the spongy bone for that matter, and release it into the blood. And here's an actual cell photograph of one of these osteoclasts. So look at all those nuclei there. I mean, this guy is kind of dizzy, right? He has a lot of nuclei going on right there, a lot of DNA. Now, the osteoblasts build bone, so they're going to do the opposite. They are going to be kind of the counterweight to the activity of the osteoclast, and they are going to do this by building bone. And so notice they have a single nucleus. I mean, these guys are just, they're just cute little cells, and we already said how they work. They're going to, they just barf out bone matrix. And so they're going to barf that out and build themselves a little lacuna, and all around you have bone ground substance, and that's what happens. Okay, so osteoclasts crush bone, they get rid of bone, they eat it up and release the calcium and the phosphate into the blood. Osteoblasts build bone by barfing out, for lack of a better term, oozing out bone matrix. Now this is kind of interesting because the initial thing that these osteoblasts are going to put out is not actually the hard bone yet. The first thing they are going to put out is a gooey substance called osteoid. So this is kind of a gooey, icky stuff. So they're going to put that gooey osteoid around. So imagine the osteoids coming along. And then after that, that osteoid kind of serves as a scaffold. So it serves as a scaffold for calcium and phosphate to deposit onto that osteoid scaffold. So it's kind of like a magnet, a gooey magnet, to hold those things. So you're going to start getting a lot of calcium and phosphate depositing on there, and that process creates a substance called hydroxyapatite. It's a mineral. It's a calcium phosphate mineral, and it's very hard, and it's also brittle. So you want just the right amount of hydroxyapatite, because too much, your bones are going to be too hard and too brittle. And in fact, there is a syndrome called brittle bone syndrome where too much hydroxyapatite makes the bones extremely brittle, and that person is constantly getting bone fractures or bone breaks. So it can actually be lethal. I actually knew a little girl that had that one. It's very, very sad. She was constantly breaking her bones. So you want a balance between too hard and too soft. And so we're going to constantly have these osteoclasts breaking down hydroxyapatite and the osteoblasts building it back up so that you maintain a homeostasis there. Well, how are we going to do all that? What is going to maintain homeostasis? So maintaining the homeostasis, well, like anything in our bodies, so much is controlled by hormones. And there are two hormones that are really important in maintaining bone homeostasis. The first one is parathyroid hormone. And parathyroid hormone is secreted by the parathyroid glands. You got 4 of them, and they're kind of in the back. They're in the back of your thyroid gland. So you'll learn about that when we get into the endocrine system. The second one is estrogen. And you're like, okay, starting to put 2 and 2 together, because parathyroid hormone controls your osteoclasts, or at least they think it does. We're not sure. I mean, this is still an area of active research. Okay, so parathyroid hormone acts on osteoclasts, and how does it do it? Well, that hormone, and remember hormones are molecules, right? They're proteins. And so that protein is going to reach osteoclasts on the cell membrane, and it is going to tell those osteoclasts to produce a special kind of substance, a protein, and we're going to call that protein rank ligand. Rank ligand. And rank ligand is going to bind to a protein called rank. So it binds to rank. And when that happens, that triggers the replication of these osteoclasts. So cell division occurs, you get more of them, and you're going to also get the activity of secreting the acid and breaking down the bone. So this causes cell division, cell reproduction, and breaking down the bone. So the more parathyroid hormone that's released into your body, the more these osteoclasts will be triggered, reproduce, and do their thing of breaking down bone. But the opposite to this is going to be estrogen. So hormone number 1 we're talking about is parathyroid hormone. Number 2 is estrogen. And so estrogen works on osteoblasts. And so what it does is it says, okay, we are having too much bone broken down. So we are going to tell the osteoblasts to produce another protein called OPG. It's a protein called OPG. And OPG acts to block the activity of rank ligand. And so if the OPG is blocking rank ligand, then the osteoclast will not be breaking down bone, and it's free for the osteoblast to reproduce and produce more bone. So this also triggers cell division and activity of osteoblasts. So you get more building of bone. So constant back and forth between building bone and breaking down bone. Now notice this is triggered by estrogen. Well, what happens to women after menopause? What happens after they age? They stop producing as much estrogen. And so the less estrogen you produce, the less you can produce OPG to block the rank ligand. And so those osteoclasts become more active than the osteoblast. You lose that balance, and suddenly your bone is constantly breaking down. It gets thin, it gets breakable. And so women after menopause are much more likely to have bone fractures, break their bones, and of course, then you run the risk of osteoporosis. Now, the lack of estrogen is one thing that contributes to the disease osteoporosis, but there's genetics involved, and of course there's lifestyle, there's diet if you don't get enough calcium in your body, and lack of exercise. And this is what I find absolutely fascinating. A lot of people don't understand this. Being sedentary is bad for your body. It's not just bad because of the, getting fat. It's bad for your bones. So let's take a look at this. Let's go back up here. These osteoblasts here, these guys are smart guys. They sit around in your bone matrix and they are constantly measuring the environment. And they know when your bone is under stress. So, when you're walking, you're running, you're lifting weights, that puts stress on the bone. But that's a good thing. As long as you're not doing too much, moderate amounts of stress on the bone is good. So bone stress in moderation is good. It is. And there's a reason for it. Because these osteoblasts register bone stress. And when they're registering that there's stress on the bone, that's telling them that, uh-oh, I better build more bone. So exercise contributes to building bone. And so people who lift moderate weights, they're going for walks all the time, they're jogging, they're constantly putting moderate stress on their bones, and their bones become stronger. So this contributes to stronger bones, and therefore less fractures, less breaks. Basically can contribute to having a longer life, because one of the main causes of things that can lead to death in elderly people is breaking hip bones. So, even in your old age, you want to be active. You want to lift moderate weights. Sitting sedentary actually inhibits osteoblast activity. Being sedentary tells the osteoclasts, we got a lot of extra bone there that we don't need. We better eat it. We better break it down. And so the more sedentary you are, the more thinner your bones get and the more brittle and breakable they are. And in fact, this is also an issue for patients that are on long-term bed rest. They're sedentary. They're not having that constant stress on their bones, including gravity for that matter. And so they actually are at risk of having their skeletal system really become very weak. And so it's important to exercise people that are on long-term bed rest so that they maintain this balance between osteoclast and osteoblast activity. So let's watch a little video clip that will sum up what we just said, because it's a little complicated, but if you think about it, it makes sense. Our body is just about balance. It's balancing everything. And by the way, I should mention, because I think I failed to mention, all the stuff that we're talking about here, where the osteoclasts are breaking bone down and the osteoblasts are rebuilding, all this is called bone remodeling. Bone remodeling. Fascinating stuff. So we are constantly remodeling our bones on a daily basis, breaking it down, building it back up, breaking it down, building it back up. Like I said, this is a busy place. This is not static. So let's watch this little video clip and see what we got.
Bone is a dynamic tissue that is continually being built, broken down, and rebuilt in a process called bone remodeling. Bone tissue is broken down and resorbed by multinucleated cells known as osteoclasts. These cells are derived from monocytes which originate within bone marrow. Osteoclasts play an important role in liberating minerals and other molecules stored within the bone matrix. Bone tissue serves as a repository for vital minerals, including calcium phosphate and various biologically active molecules, such as growth factors. The release of calcium from the bone can play a role in maintaining its homeostasis within the body. The cells responsible for building new bone tissue are known as osteoblasts. Osteoblasts are thought to be derived from cells found to be associated with blood vessels. Osteoid, which is predominantly made of collagen. Minerals start to crystallize around the collagen scaffold to form hydroxyapatite, the major inorganic constituent of bone, which contains calcium phosphate. Bone mineral density, or BMD, can be used to estimate the strength of bone and to assess the risk of fracture. As osteoblasts form new bone tissue, many become embedded within the matrix and differentiate into osteocytes. The structure, composition, and cellular processes that occur within bone allow it to simultaneously serve as a calcium reservoir. The skeleton changes across the human lifespan. This is characterized predominantly by bone formation and growth throughout childhood, followed by a gradual loss of bone density that begins in early adulthood that can accelerate significantly in older adults. The density of bone is modulated by a group of cells including osteoclasts, which are multinucleated cells that resorb bone, and osteoblasts, which refill the resorption cavities created by osteoclasts. Osteoclasts anchor themselves to the surface of bone. This creates a microenvironment underneath the cell, which is referred to as the sealed zone. Within this zone, the osteoclasts create an acidic environment that dissolves the bone's mineral content. Once the mineral content of the bone has been dissolved, enzymes released from osteoclasts remove the remaining collagenous bone matrix to complete the process of resorption. Following resorption, osteoblasts move into the resorption space and start to produce and deposit organic matrix called osteoid. Osteoid, a substance made predominantly of collagen, forms a scaffold in which minerals including calcium and phosphate begin to crystallize. Some active osteoblasts become trapped within the matrix they secrete and thereby become osteocytes. Other osteoblasts will undergo apoptosis or will revert back to lining cells which cover the surface of bone. This cycle of bone resorption and formation is referred to as remodeling. There is also a process where bone formation by osteoblasts occur without prior bone resorption by osteoclasts. This results in an increase in bone mass and is referred to as bone modeling. Bone modeling promotes the growth of bones and is important for maintaining bone strength. Remodeling also plays an important role during bone growth by optimizing the growing structure. After the age of 30, most people experience a gradual loss in bone mass due to a relative decrease in the activity of osteoblasts compared with osteoclasts. However, there are many factors that impact the process of bone remodeling and influence the degree of bone loss we experience as we age. For example, medications such as glucocorticoids, which can promote osteoclast activity and also reduce bone formation. Proper nutrition and physical activity can help strengthen bone. It is also believed that osteocytes form a complex network in bone that can sense any increased workload on the bone and respond by triggering the differentiation and activity of osteoblasts to increase bone density. Conversely, when bone experiences reduced loading conditions, such as during long-term bed rest, resorption and remodeling increase to eliminate underloaded bone. Loss of bone mass reduces its strength and increases the risk of fracture. This highlights the importance of staying active, maintaining good nutrition throughout life, and being aware of personal risk factors associated with low bone density. Osteoclasts and their precursor cells are regulated by different signaling pathways and molecules. One key regulatory molecule is the protein rank ligand, which is produced by a variety of cell types, including osteoblasts and their precursor cells. This protein has been shown to exert its effects on osteoclasts and their precursors through an interaction with the cell surface receptor, rank. The interaction between rank ligand and its receptor, rank, triggers the activation of signaling pathways, which are involved in the differentiation, activation, and survival of osteoclasts. Such signaling pathways can trigger the fusion of precursor cells to form mature multinucleated osteoclasts. On these mature cells, rank ligand can interact with its receptor, promoting osteoclast activity and survival, leading to increased resorption of bone. To balance the effect of rank ligand, osteoprotegerin, or OPG, is secreted from osteoblasts. This protein acts like a decoy in binding to rank ligand, preventing rank ligand from interacting with and activating its receptor on the surface of osteoclasts. Various hormones and cytokines modulate the balance between rank ligand and OPG. For example, parathyroid hormone stimulates the production of rank ligand while at the same time repressing the expression of OPG, an effect thought to increase the activity of osteoclasts. Conversely, estrogen can increase the expression of OPG and inhibits rank ligand signaling. These changes, which are thought to reduce the activity of osteoclasts, may contribute to estrogen's ability to preserve or increase skeletal mass.
How cool is all that? So hopefully that helps you to kind of understand the amazing physiological mechanisms that underlie being able to have these bones. I mean, we are having a dynamic place in these bones, constant breaking down, reforming, and that is known as remodeling. Now it's time to go on to learning about exactly how bones grow from the time you're an itty bitty little mini baby in your mama's tummy on up to when you are an adult. So let's take a look at some of that, shall we? First kind of bone growth we're going to talk about involves flat bones. So this is flat bone growth. And you guys will remember that flat bones are things, they're flat bones, like your scapulae, like the bones of your cranial cap, your frontal bone, and your parietal bones, and your occipital bones, etc. So basically periosteum and compact bone with not much spongy bone in the middle. So those are your flat bones, and the name for this kind of flat bone growth is called intramembranous. That means within a membrane, intramembranous ossification means bone growth. Ossification. So intramembranous ossification is bone growth of your flat bones. Okay, so intramembranous ossification, and basically this is going to start when you're a little itty bitty mini baby. We're talking about 8 weeks or so of embryonic development. 8 weeks or so of embryonic development. So at the time of this recording, I happen to be pregnant and have a cute little 12.5-week-old fetus moving around in my belly. So she or he, whatever the case may be, already has some bone, flat bone forming, because this started a few weeks ago at 8 weeks. Well, what we have to do to start the process of producing bone is, when you're talking about flat bone growth, this will be different from the long bone growth we'll talk about in a minute. When you're talking about flat bone growth, that is a tongue twister, flat bone growth, you're talking about the need for a connective tissue scaffolding to be there at the beginning. So a TC scaffolding, and the kind of connective tissue that is going to sort of serve as the scaffolding, or we'll call it the model, it's a connective tissue model. You might recall the name of this connective tissue, dense irregular connective tissue. Dense irregular connective tissue. Where else do you find dense irregular connective tissue? The dermis of your skin. You remember all that? Absolutely you do. Of course you do. But all mini babies, embryos, are going to start off with the formation, once they've actually started taking on form, and they're not just little cells anymore, they're going to get the deposition of dense irregular connective tissue. And this is going to serve as our model for the production of flat bones. So just like with any bone, we're going to need some stem cells here. And so you guys remember that the stem cells for the creation of bone are called mesenchymal cells. Mesenchymal cells. So these are your stem cells. They're undifferentiated. They haven't become what kind of cells they're going to become yet. And if the right conditions are there and we need to form bone, these guys are going to start replicating and they're going to start differentiating. And eventually they're going to become osteoblasts. And again, this is at about 8 weeks of embryonic development. Now, where this is going to occur on the embryo is in a place called the ossification center. Ossification center. And often the ossification center would be kind of like in the middle of your flat bone. So for example, if I take back my bone here, so imagine, here's what's going to become the parietal bone. The ossification center, where these mesenchymal cells are starting to kind of conglomerate, it's going to be right in the center of that dense irregular connective tissue. And so they're going to start accumulating, kind of forming a mass, becoming osteoblasts, and from there, you guys know what happens once you get osteoblasts there. These osteoblasts are going to start spitting out bone matrix, right? And so once again, you have your little osteoblast is going to start spitting out bone matrix, so calcium phosphate and other things, and form himself a little lacuna around himself, and now we're getting bone matrix over here. So that is your bone matrix. Bone. I just love that word, bone matrix. Reminds me of the movie, The Matrix. So we all know what we call it once that osteoblast is in the lacuna. We know that it is now an osteocyte, right? Okay, so now we have our osteocyte, and we're forming this bone matrix around it. But the first thing that's going to happen is these guys are going to form, actually from the cytoplasm, little processes. So you'll learn later a process is like a little stick. It's like sticking out. And these things are going to be called spicules. So this osteocyte is going to start forming spicules. And as these spicules get larger and larger, they eventually will become your trabeculae. So your trabeculae, as we know, are the basis of forming your spongy bone. And so in some cases, you might only get spongy bone, but in most cases, as the spongy bone grows and grows and grows, these osteoblasts are dividing and reproducing, so you're getting more and more bone matrix laid down. Eventually, it may just grow out until the outer parts become compact bone. So from there, on the outside, some of these may become compact bone. So basically what you're getting is, imagine that you have, all these spongy bone forming in here. And as the cells on the outside, the osteoblasts, start reproducing, spitting out more bone matrix, boop, 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 eventually you start to get compact bone forming. So there's our compact bone, and it's going to start by the formation of the periosteum, which is on the outside. So, on the very outside, you're going to have your periosteum. So that, in a nutshell, is intramembranous ossification. Again, this occurs during embryonic development. It occurs at an ossification center where mesenchymal cells differentiate into osteoblasts, differentiate into osteocytes through the production of bone matrix, forming spongy bone, and eventually the periosteum with compact bone under it. So this happens during embryonic development, and you need a model. You need a CT, a connective tissue model of dense irregular connective tissue in order for this to happen. And again, this is all for flat bone growth. We're talking about flat bone growth. Now, the next thing to look at is long bone growth. So let's join me. Okay, so now you're familiar with the way flat bones grow through intramembranous ossification. But what about these guys? What about these long bones? How do these guys grow when we're little mini babies in our mommy's tummy? Well, they're going to use a different process. And the process is actually a little more complicated than with flat bones, but at least as interesting. Very, very interesting. So with this, we're going to be talking about long bone growth. Long bone growth. And the name for this long bone growth is called endochondral ossification. Of course, ossification means bone formation. So, wow, that was a horrible line. Oh well. Endochondral ossification. Endo means on the inside. Chond means cartilage. You already know that. Inside the cartilage. It gives you a clue into the kind of growth we're talking about here. So you already know about cartilage. And in fact, with intramembranous ossification of flat bones, we were dealing with dense irregular connective tissue with the ossification center occurring kind of in the middle of the area and then growing out. Well, long bone formation is different, but we're still talking about baby embryos. So these embryos, about maybe 9 weeks development or so, 8-9 weeks, I mean, it's all relative. At this point, all long bones of embryos are made of hyaline cartilage. So these are hyaline cartilage. And so we're going to be using a hyaline cartilage model to create these long bones. Now, how is this going to happen? Well, let's go ahead and make myself here a little long bone. Now, you know me and drawing, but let's make kind of a long bone. See, long bone? Here's our long bone. But this is not really a long bone because this bone is not bone at all. It's made of cartilage. So it's got our little, chondrocytes here and their lacunae. It's hyaline cartilage. That's what at this stage, this embryo's long bones are made of hyaline cartilage. But we don't want to keep hyaline cartilage. We want to turn this into actual bone. Now, okay, so we know that this long bone is made of hyaline cartilage, and on the outside of it is kind of like a periosteum, but it's not a periosteum. It's a perichondrium because it's cartilage. So I'm going to make this little perichondrial sheath right here. So this is the perichondrium. Now, this perichondrium eventually starts to get replaced by periosteum, and it starts here. So this is forming periosteum. So it is actually forming bone because now we have some osteoblasts here, and they're starting to lay down that bone matrix. So we got periosteum, and this is starting to become an issue for these poor little chondrocytes there because now they're getting cut off from diffusion. Remember that hyaline cartilage chondrocytes can only be maintained through diffusion because there's no vascular tissue in there. There's no blood. And so they rely on diffusion to stay alive for their nutrients in their water. But now you got this bony thing going around the shaft, and this is called, lo and behold, very technical term here, a bony collar. And it's produced by the periosteum. And anywhere where this bony collar is, these chondrocytes are in big doo-doo because they can't get diffusion in there. And so the only way they're going to stay alive is if they can get some help. Well, lo and behold, this perichondrium will start to kind of come quasi to their aid. You'll get little projections starting to form from the periosteum inward. And these projections are called, they're buddies, right? They're buddies to the chondrocytes, periosteal buds. Okay, so that's a periosteal bud. So he's kind of coming to the rescue, bringing blood vessels, but along with them, he's bringing his buddies, osteoblasts, or what will become osteoblasts, and osteoclasts. You guys know these. Well, so the osteoblasts are going to start forming bone. They do what they do, right? So let's get our little osteoblasts in there, and they start forming their own little lacuna. So now you're starting to get bone matrix forming. So that's bone. And further alienating these poor little chondrocytes. They are really not doing good now. And so what do? Well, what do all good chondrocytes that can't get a good blood supply going do? They die. Our chondrocytes die. And when they die, some interesting stuff happens. Because as these baby chondrocytes die, well, they're not really babies anymore. Ooh, what was that? That's an arrow. Okay, there we go. So as these chondrocytes die, they hypertrophy, they enlarge. And in the process, as they're dying, they kind of like ooze out this hard substance. So they're going to ooze out calcified cartilage matrix. So they are oozing out calcified cartilage matrix. So that's hard stuff, but you don't want it because it's kind of competing with the bone matrix that the osteoblasts are trying to lay down. So we want to get rid of those guys. And fortunately, we can do that because we have osteoclasts in there. And what do osteoclasts like to do? Remember, these are the multinucleated monster-looking things, right? They're going to come along and they are going to eat this calcified cartilage matrix. So osteoblasts are putting out bone matrix while these osteoclasts are munching up this calcified cartilage matrix that the chondrocytes, dying chondrocytes, are producing. And so the net effect of this is you start getting a kind of a hollowing out of the middle. That's the formation of our medullary cavity because these osteoclasts are kind of eating up all the old stuff. And meanwhile, the good little osteoblasts are producing more bone matrix. And it's going to become spongy bone initially. And meanwhile, what are these poor little chondrocytes doing? Well, those that didn't die still survive But they are only going to survive in 2 places. Well, initially as embryos, as fetuses, there'll actually be hyaline cartilage still in all these places. But shortly before birth, a secondary ossification center will occur about right here, and in bones like the femur right here. Some bones it'll only be one side, but that will become the secondary ossification center. And I know you actually can't see that with my picture there, so that'll become your secondary ossification center. And that happens shortly before birth. So eventually all this will also become spongy bone. So this will become spongy bone, and all this was spongy bone. And then we know eventually we'll get compact bone on the outside, because eventually our periosteum will go all the way around. Whoop, right? Now we've created nice ossified bone here. And so the only place that the cartilage is going to remain is in these epiphyseal plates. So let's see, I'm kind of running out of room here. Let's see if I can do these. So that right there, and that right there, this is an epiphyseal plate. That is your growth plate. And those growth plates, those epiphyseal plates, will remain at the ends of your long bones well into adulthood. In fact, till you're about 30 or so. And they'll keep growing till then. And in fact, they don't turn into bone until you're like around 50 or so. I mean, it depends on the person, but those will be the sites of continued growth, elongation. That's how your long bones continue to grow when you're a child. And so those are called epiphyseal plates. Now let's take a look at some of them because they're really, really cool. I got a picture right here. Check this out. So this is one epiphysis, so one end of a long bone. So we're looking, for example, this is probably humorous here, but we're looking at one end. And so what you see here, in here is still your spongy bone. And over here is more spongy bone. But right here in the middle, all this thing right here, that is your epiphyseal plate. Well, not yours, but whoever this person's was. Epiphyseal plate. So that is your growth plate, and that's where you'll continue to grow to elongate your bones. Pretty cool, huh? So that is, in general, endochondral ossification. And so now what we're going to do is we're going to learn about the different ways that these bones grow after birth, including how these epiphyseal plates work to actually elongate that bone. So that's the next topic. Okay, so let's talk about these 2 ways that we can get growth in bones after birth, for example. So 2 forms here. We're going to talk about appositional growth. And we're going to talk about, and I'll move that down because I know my face is probably blocking that. So appositional growth versus interstitial growth. Positional growth versus interstitial growth. Let's draw a line so we can compare and contrast. Now, appositional growth is kind of like growing, I sort of think of it as like rings on a tree. So imagine this is your long bone here, and we of course have your periosteum on the outside of your long bone. And of course, what I'm talking about right now is for bone growth, but actually the same sort of things happening when you're dealing with a perichondrium as well. This is surface growth. This is growth that happens at the surface of a bone. So this is how the bone is increasing in diameter. So that's the key there. This is diameter growth. So this is how bones increase in diameter. Diameter is the key here. So not length, diameter is how they increase in girth. They get wider. And it's pretty simple, actually. Basically, you're going to have a bunch of osteoblasts lining by the periosteum here. And with the right signals, these guys will start cell division. So they start dividing. So the more and these are osteoblasts, So the more cell division you have, the more osteoblast activity you can have. And again, what is osteoblast activity? Osteoblasts forming themselves a nice little lacuna as they barf out bone matrix. And so this is going to lead to the formation of osteons. So formation of osteons. Now, if this doesn't make a whole lot of sense to you, no worries. I got a video to show you. So hang on one sec. Let's get that video.
Bones grow through the process of appositional growth, the formation of new bone on the surface of older bone or cartilage. Osteoblasts beneath the periosteum lay down bone to form ridges around a blood vessel. The blood vessel lies in a groove between the ridges. The groove is transformed into a tunnel when the bone built on the adjacent ridges meets. The periosteum of the groove becomes the endosteum of the tunnel. Osteoblasts from the endosteum lay down bone to form a new concentric lamella. The production of additional concentric lamellae fills in the tunnel and completes the formation of a new osteon.
Again, these osteoblasts that are lying under the periosteum are going to start laying down bone matrix, and eventually the matrix is getting laid down in lamellae, and eventually you have your osteons. So it's all about osteon formation for increasing the diameter of the bone. Now let's contrast this with interstitial growth. Interstitial growth, we're talking about elongation, kind of like we just talked about. But there's really two categories of this. You have how this happens in cartilage and how this happens in bone. And this is kind of a misnomer, because even with bone, it's not actually happening in the bone. It's happening with those epiphyseal plates, which are made out of cartilage. So there really is no interstitial bone growth, even though they'll tell you there is. It's all about cartilage. Well, how does this happen? You already know how this happens. I mean, you already understand totally about how cartilage will produce more cartilage, right? So if we take our, little chondroblast, and we know that the chondroblast will start producing cartilage matrix to form a lacuna, and there's cartilage matrix all around it, and then it's trapped in there. Well, the more of this that happens, the more you produce. And in fact, sometimes these guys will duplicate, and so you get even more produced. But that is generally what's going to happen to produce more hyaline cartilage in a long bone. Well, we already saw what happens with elongating bone on top of that cartilage, but after birth, it's going to take place in the epiphyseal plates. So it's taking place in those epiphyseal plates. And so now what we want to do is take a deeper look at what's going on in those epiphyseal plates to lay down more bone to make those bones. Okay, so now we're at the epiphyseal plate, and we're going to see what happens at it. And so just a reminder, you're at the epiphysis end of a long bone, and your epiphyseal plate is like right there, and it's all hyaline cartilage. So just don't forget that when we're talking about epiphyseal plates, we are talking about hyaline cartilage surrounded by spongy bone. And this is your growth plate. So epiphyseal plate is your growth plate. This is how your long bones are going to continue to grow until you're an adult. So this is your growth plate. Eventually, your growth plates will close and become ossified, but in children, they're still very, very much cartilaginous. And so the way this is going to happen is you're actually going to just basically get cartilage changing shape. These chondrocytes will start changing shape, changing activity, until they produce that calcified cartilage matrix that we talked about before. So how does this happen? Well, in the furthest away from the area where the calcium is going to be deposited and the new bone growth is going to be deposited, you have this region up here, and this is called your resting zone. So this is your zone of resting cartilage. Means it's not doing very much, just kind of hanging out. So that's your zone of resting cartilage. But once this guy gets ready to grow, it's triggered by hormones, growth hormones, and etc. all of a sudden you can see that these chondrocytes that were up here are starting to change. They're elongating. Look at that. They're elongating and they're multiplying within these little lacunae. So the lacunae are actually elongating and multiplying the cells. So the lacunae, and I should actually write this, that's an elongated lacuna. They're starting to elongate and the cells are starting to proliferate. And so this is, because the cells are proliferating, this is your zone of proliferation. Ah, makes sense, right? We're proliferating chondrocytes, therefore it's the zone of proliferation. And then, as these guys are proliferating, they're moving down, and what's happening to the lacunae? They're getting big. They're hypertrophying, if that's a word, hypertrophying. You already remember that hypertrophy of these chondrocytes is happening when they're about to die because of the bony collar. They're starting to get cut off from diffusion, and so these guys are going to start secreting calcium. And so this is called your zone of hypertrophy. Hypertrophy. Hyper means a lot or big or something like that, and trophy means eat. So they're kind of big eaters. But in a sense, it just means they're getting big. They're getting big, they're getting old. They are ready to die. They've been cut off from their nutrient supply. And so by the time they're down here, they have just died and oozed calcium. And so this is your zone of calcified cartilage. Calcified cartilage matrix. And what happens after that? We know what happens to that. Osteoclasts are going to come along and start eating away that cartilage, and osteoblasts will replace it with bone. So this is happening at either end of this long bone, right? We have these epiphyseal plates. These guys, these chondrocytes, go from an area where they're resting and happy and getting what they need, their nutrients, to an area where they're proliferating with the lacunae elongating and the chondrocytes replicating, to an area where they're starting to die because they're hypertrophy and they're cut off from their supplies, and boom, they just squirt out this calcium deposit, which will get eaten by osteoclasts, and the cartilage will be replaced by bone by the osteoblasts. So that is epiphyseal growth. It's a kind of endochondral growth of the long bone, but it's the long bone growth that continues all the way until you're an adult. All right, I hope you have enjoyed this, learning about the remarkableness of what makes up bone, the bone tissues. And now you're ready to actually go and learn your bones, which is what everybody thinks about learning in an anatomy class. All right, enjoy.